our not reading. Can you can. Wait a minute.
Hello. So shall I start?
Yeah, you can start.
Okay. So I guess I'm going to talk a little bit about defense. That maybe not the most relevant for the moment, but about how cells defend themselves from invaders. So most of you probably have been introduced at some stage to what's called the central dogma of molecular biology. So you've got DNA there. What does DNA do? Oh, and this is a dialogue, by the way, not a lecture. If I was giving a lecture, you would all be asleep now. So what does DNA do? If you were a computer, what would be the equivalent of the DNA for the computer? All of you have computers or phones in front of you. So what's the computer manages information, a cell manages information. So what's the equivalent of the DNA in the computer?
I suppose that in the computer, DNA would be the, hardware, like something that stores information about it.
Right. So the long-term storage of information would be the hard drive in a computer and the DNA. So it's where you keep information long-term. Now, in a cell, you actually, in a computer, you change that information. In a cell, the original view of it was that you really don't change that information yet. And we have been, I would say, adjusting that view of the world since Watson and Crick to the idea that one does change the information under various circumstances. But given that as it may, the most stable form of information in a cell is DNA. But a hard drive doesn't do, if you just had a hard drive, you would not have a computer. And so you need something that will act on that information. And in fact, at any given time, there's some information that is really active and being used. And so in a computer, what is that? So what you have a hard drive in your computer, which is the long-term storage. When you turn off the computer, that information is maintained. What's the short-term memory for a computer? Where do you keep information in the short term? So those are your memory chips. They only keep the information for the short term, and they hold what's important in being used at any given time. So in the cell, what's the shorter term memory?
I suppose it's RNA.
Yeah, so it's RNA. Okay. So a cell does more than a computer. My computer, if I put it in the middle of the garden and I asked what would happen if I came back, there would not be more computers there. There would be a very rusted computer after a while. And so it doesn't duplicate itself. And people build computers. Computers don't, for the most part, build computers, although there is computers involved in that process. And so a cell is different, right? Because a cell, in principle, cells can be a part of rebuilding themselves, although usually it's a much more complex structure. So our cells have the ability to rebuild tissues, to do specialized tasks, and ourselves, our organisms, are able to rebuild, to build more of us, in particular, through very complex processes. So if that all goes well, then any given organism will continue to proliferate. So if I put a computer in the backyard, it will keep dividing I mean, it won't divide, it'll stay there. If I put a cockroach or an ant in the backyard and it eats the plants, what are you going to get? You're going to get more cockroaches or more ants. And so you do have biology moving forward and creating more of any given species. You have as Darwin described it, a survival of the fittest in that sense, but also a survival of the species that can most effectively duplicate itself. So in that world, you place your need to move forward as biology, as organisms, in the context of, well, okay, so what you need to do is replicate and not die. Okay, so that all is fine and good. And now let's layer on top of the fact of those things, the fact that we don't live alone in our environment. We also share it with other organisms. And under the best circumstances, of course, the organisms will coexist well and each provide something to each other from the environment and move forward, and biology eventually moves forward and converts the world to a lot of living things. And that's a positive thing. So what happens in that kind of environment if you don't have any control on what the individual organisms do? You have a benefit to any given, there's a hand raised, okay. Do you want to?
Maybe the whole environment will be in the environment will be collapsed.
Okay, so that's one of the things that can happen, is that biology is so effective that the duplication of individuals and the propagation leads to too much, to overuse of resources, to overuse of space, and as you say, things collapse. So that can be one end of a given state in biology. Now, what happens in that case, of course, is not that the whole thing necessarily collapses, but that you have an approach towards, instead of unfettered, unblocked growth, you go to a stage where there's an equilibrium, where the environment can take some additional organisms, especially if some are being lost or dying, but that's limited. And so you have some limitations that occur on the biological growth of something. Because if you have twice as many cockroaches every week, then after a year you'd have so many cockroaches that they would cover the whole earth many times over. You couldn't do that. And so at some point you run into a situation where things need to be limited. And in some cases that happens through the biology of the organism, because the fitness of the organism depends on its ability to limit the use of resources. In some cases, there is an ability of organisms to self-regulate. But if that doesn't happen, what occurs? Before you take up all the resources, what happens? So you have a whole bunch of cockroaches in the backyard. There's suddenly fewer and fewer resources. What would occur?
These cockroaches will gradually either die out or will displace themselves in the place with more resources to maintain their life.
Right. But if you don't give them more resources and you don't, the ones that are not dying, what is there going to be there? Which ones will be the ones that survive that? Go ahead.
The one who will be eating others.
Yeah, or at least it would at least be grabbing resources from others, and so you end up with a environment that selects for selfish behavior, basically. So in the organismal world, that occurs all the time, of course. And then you have species and organisms that try to counteract that so that they can do their own biological life. But it is something that goes on. That also happens at the microscopic scale in terms of individual genetic elements, genes, as we would call them. And most genes in an organism are part of the beneficial adaptive component of that organism, and they benefit the organism and therefore benefit themselves. And so if a gene, you have 20,000 or so genes, or maybe more, and each of those genes has the potential to contribute to your fitness, to your happiness also. And if they all work together well, that makes you a more fit organism. And that can happen also with an ecosystem or a species, is that to the benefit of the species, for the benefit of the ecosystem, things can work together. But genes also can go awry in the sense that they suddenly become selfish and they want to make more of themselves as a gene at that stage, and to try and propagate, not to be too much of an what we call anthropomorphizing, which is to put onto inanimate dead objects the features of human beings. But there are genes, there are bits of DNA which are selfish and that mostly try and make more of themselves rather than trying to contribute to their host, contribute to the needs of the community. And so how does one, if that was, okay, so what do you call that kind of gene?
You mean the gene that is sort of selfish?
Yeah. So it's a piece of DNA that instead of contributing to its host, contributing to the organism it's in, it is mostly trying to make more of itself.
I have no answer.
Thoughts? So I would call that a virus. So essentially what happens when you open up, when you have this broad environment where there's resources that are present from an organism which has built itself with metabolites, with molecules of life, like sugar and fat and good things like that, proteins. If a gene only has the ability to duplicate itself in that environment, then it will have the ability to propagate, and there could be a lot of it. Now, in the real world, viruses are usually more than one gene. They will be a number of genes, but a relatively small number that working together can make a lot more of the initial material. And so, for instance, the very simplest viruses may only be able to produce one product, and that product is an enzyme that makes more of the DNA that started with, or the RNA. But usually there'll be many. And so viruses can be either DNA, in which case they're going to duplicate themselves as DNA, or RNA. And so I began to study this field, not deliberately, but because I was trying to be a developmental biologist. I grew up here in the US, although my family came from not too far from where you guys are right now. But I grew up here in the US, and I came to England as a postdoctoral fellow, which was a wonderful thing to do, to be able to go to another country, study science, learn a little bit of the culture. And I'm really poor at languages, as you might guess. And so I was happy to be in England, where they didn't quite speak the same language, but I managed to understand some of what people are saying. And so what I wanted to study was developmental biology. And I had this great idea, which many people had at the same time, that we could figure out the relationship between the DNA present in the genome, essentially the information on that hard drive, and the beautiful structures that form in biology, whether it's a plant or a person or a worm. And if you wanted to do a one-to-one correspondence between genes and structures and parts of the structure that you see, the idea was you could manipulate the gene and then see what the consequences were. And so the dream experiment 35 years ago, when I was finishing up my graduate study, 45 years ago, when I was finishing up my graduate studies, was that one would take a individual organism and try turning off every gene individually and see how that affected what the organism looked like. So if you have a lizard and you turn off one gene at a time, which of the genes affects the fingers, which of them affects the scales, which of them affects the stomach? And that catalog seemed to me, and a lot of other people at the same time and since then, to be a starting point for figuring out how you build complex structures. And so this is still working in the world of the organism and its genome, a bunch of genes that work together, because that's how they have evolved to build this organism, to build this lizard or whatever it is. We were working with a worm, in fact. And so the kinds of experiments that I wanted to be able to do were experiments where we would put in extra copies of a gene and see what that did, or take away copies of a gene and see what that did. And so that was the kind of experiment that we set up to do. And those experiments didn't work the way we expected them to. So we would try and put in extra copies of a gene and it would shut down instead of turning on. Or we would try and take away copies of a gene and we couldn't get those things to work very well, those experiments. And so we had this goal, which was as trying to be the director in this play and change what the actors were doing and see what happened to the outcome of the play. And the problem was that when we did that, often what we wanted to happen didn't happen. And so if you think about it from the organism's point of view, and we hadn't at that point, we weren't really thinking about that. Others who were in the same field were, but we were just thinking about it in terms of very simple, we want to do this experiment, how do we get it done? And it wasn't working the way we thought it would. The insight there that was really critical was that those experiments were really not experiments to study developmental biology. They were actually experiments to study the response to viruses, because we were trying to put extra DNA in a cell and see what would happen, or take away DNA that would normally be there. And those are all things that viruses do as part of their life cycle in order to hijack a cell. And so in a rational world, what would happen is that some of the resources in the cell would be dedicated not to building a new cell, but to making sure that the darn virus next door doesn't come here and mess things up. And so trying to get at those mechanisms of how a cell recognizes when something is trying to mess it up and intervenes so that it doesn't get messed up is something that has been most of the rest of my career, and many, many other people are working on this also. And so each of our cells, in addition to having a machinery to make more of that cell, has a bunch of machinery to avoid viruses, to avoid any kind of selfish information, because a lot of what one does to move forward in biology is to avoid interference, hijacking, various other things from genes that have gone rogue, have gone into a mode where they're trying to mess things up. And so the question is, well, how do you set that up? So taking again from the cell's perspective, which again, I was very late to do as a scientist, but from the cell's perspective, how do you set up a mechanism to protect yourself from selfish genes? And so any thoughts on how you would set that up?
Well, you know, maybe form some preventative genes that would preclude this interference.
Mhm. Right. So we're gonna take up some of our information content and some of our energy in trying to prevent that. And you said you formed some genes that will do this. Now the question is, how do you recognize the difference between the virus that's trying to replicate itself and make more of itself, and just normal gene expression, normal cellular processes that are needed to survive and to duplicate the organism? And that's the really hard part. But you do need sort of two different things. One is a recognition for what's viral activity that you want to shut down. And the second is something where you can then protect yourself from it. So let's take those in order. How do you recognize when there's a virus trying to replicate itself? And there's no wrong answers to this. There are many right answers to it, but yeah.
Well, I think there is sort of recognition of the receptors on the virus's membrane. So they are different from our cells' membranes.
Right. So that's a great idea. So we're going to have a database of all of the viruses that have ever been encountered and their characteristics. And we're going to teach our immune system to recognize all of those different receptors or various surface proteins on viruses, and to eventually we'll get rid of them, destroy them, whatever, but that can be our trigger. So we do some of that. And if it's a really nasty virus, then if that virus over time has challenged our species, then we will eventually evolve some way to recognize that surface protein and go after the virus that way. What happens if we do that? What happens to the virus? The virus also is subject to evolution and to selection for the adaptive, the survival of the fittest. So what would happen to the virus if we had a really good way of recognizing its surface protein?
Well, I think in this case, it would never been able to enter our cells. And so that when we started biology in our school, it would say that viruses is also, it helps to evolve organisms. And when it enters our cells, it can insert some of its RNA in our DNA, and this way make changes.
Yeah, I mean, some viruses would be able to do that. But one of the things that would happen if we were very, very good and very, very effective at recognizing one specific outer protein of a virus, that virus would have every reason to start because mutations always occur in biology, the virus would have an advantage if it mutated that protein to no longer be recognized. So we're creating a strong selective environment, which we do with viruses in the real world, for something that would mutate to no longer be recognized. So it would protect us somewhat, but eventually the virus would be selected to start mutating and eventually get away from that recognition. And so it's definitely beneficial for us to have what we call a repertoire of different components that can recognize specific viruses that might be a problem, but eventually they will start to mutate to get away from that. So what we're talking about so far is a static immune response, a fixed immune response, which we have against certain things. And there probably are viruses and bacteria, maybe more significantly, that are prevalent enough in the environment and have been around for a long enough time that it really is to our benefit to have a fixed mechanism there to attack them. But in order to deal with the real world where the viruses are going to be mutating, it helps to have a more flexible way to deal with it. And so if you could build a system that instead of having a fixed recognition of one specific protein that was on the surface of one virus, could build new recognition as it needed, that would be great. Now, so some of us, so maybe you're all familiar, I'm sure, given the pictures with COVID and coronaviruses. And so this virus infected the human race called coronavirus. And then we developed a vaccine to it. We didn't develop, I had nothing to do with it, but the human race developed many different vaccines to it, which involve priming the immune system to recognize the surface protein of the virus. What's been, and at the same time, a lot of people were infected and those people got their own immunity. So what happens to the virus under those circumstances, of course, is that the virus mutates, which it has to be alpha, beta, Omicron, BA 4.5, all these things come up and they are now capable of evading the immune response, not because the virus has necessarily gotten any smarter, but because the ones that the viral infections, the viral populations that have survived are the ones that were able to evade the antibody response that we got. And so, but then what do we do? Well, hopefully what we're doing now, I think, maybe, is building a new vaccine that can deal with the new viral variants. And so in our own biology, and in part of this whole thing, actually part of the same process, we're doing the same thing, is that we are elaborating responses. If a virus changes, we're elaborating responses to that virus. But that's a complicated thing. How do you know if you have a flexible response system that can say, hey, there's this thing that's bothering me, make an immune response to it? How do you avoid making an immune response to your own molecules, which would be a problem? That would be what's called an autoimmune response, where you attack yourself. And so how do you prevent the biology that's supposed to protect you from actually causing problems?
Well, probably to remember the proteins your cells have, the different kinds of cells have.
Right. So if you were able to, when you're building the immune system to start with, if you're able to take the first challenges that come up and say, well, actually, this must be self. And so if in a very young organism, if you have a whole bunch of random antibodies or T cell receptors, which are the recognition parts, and you get rid of the ones that can recognize self early on, then you're only left with the things, they may not do any good, but they're not going to do as much harm. And so if you can get rid of the self-attacking antibodies originally and have everything else hanging around, that's good. But that doesn't quite get you an immune system. You also have to figure out what you then respond to otherwise. And so if anything foreign triggers a response, triggers an expansion of the cells that could attack that thing, then you might have an immune response. But in fact, our bodies do go one further on that, in the sense that they only trigger an immune response if associated with the molecule they see is some kind of activity, some kind of indication that it's also problematic. And so if you were to just purify the surface protein of COVID as a very pure molecule and inject it into people, you would not have a very effective vaccine, because your body would see it and say, oh, this is probably something foreign, but I don't know if it's something really bad. What is more effective and what vaccines are made of is a mixture of the specific component, like the cell surface protein of coronavirus, and some kind of what's called an adjuvant, which is a non-specific molecule that says, hey, there's a problem. It's an irritant, basically. And if you mix irritation with something, then that becomes a logically much more rational way to trigger an immune response, because you're only going to trigger a response if there's a combination of something new and then some destruction of our own cells. And the combination of those two things says, let's really go after that. And so you don't want to be aggressive towards any stranger because you're more likely to end up attacking something that is really part of your own system there. You want to attack something where there's a juxtaposition in the same place of something new and some kind of self-destructive characteristics. And so the immune system, the classic immune system with antibodies and T cells, has a dual recognition which it takes to start going. One is this foreign component thing where it has antigen receptors which are selected not to recognize self and therefore can recognize other things. And then combined with that is this requirement to see untoward effects, to see adjuvant behavior, to see irritation. And so that's one of the ways that the classic immune system works. And so when you get a viral infection, you probably don't respond particularly quickly to the fact that there's this foreign virus circulating in your blood, even, or in your nose or whatever. That's going to happen anyway. What you respond to is the juxtaposition of the foreign virus information plus really sick, unhappy cells, which are sending distress calls out. I'm sick. And so a lot of the function of immunology is, of course, the recognition of foreign things, but a lot of it also is how do you decide to respond with a danger signal if the cell is sick, and to use that information to prime a response, which would say, okay, now really go after this molecule that's new here. Okay, so that is my very naive introduction to classical immunology. And I have to tell you, as a graduate student, again, 45 or so years ago, I learned immunology from some of the people that were experts in the field. And then about 20 years later, I was actually, strangely enough, I got an F in the immunology class. It was a long story, but I failed the class, but I still graduated from graduate school. But then about 20 years later, for various reasons, I was teaching immunology at Johns Hopkins, where I was at the time. And I found that my notes from 20 years earlier were completely useless. The faculty that taught it were wonderful, but the field had changed completely in those 20 years. And then that was also about 20 years ago. I'm an old guy. So I have gone back occasionally and looked at those notes from the early 2000s. And immunology has again changed rather substantially since then. So immunology is also a field of science that changes relatively rapidly. All of which is to say, we don't really understand these things all that well. We don't understand how the immune system really makes that large variety of recognition molecules that can be selected from to get things that are useful, and how it selects which ones to make, and how it avoids self-responses. Although we know a little bit about that, and we have models for all these things now. My naive understanding is that there's a lot of questions out there, and they're important questions because they will determine how we get, to begin with, at vaccines against viruses. And then, by the way, one of the things that can happen that's bad also is cancer. And again, that's a population of cells in that case, which gets selected for, whose adaptation is selected for, that we don't want. One of the approaches to deal with cancer is vaccines. That has begun to be a really effective protocol, but we don't know how that works either. And there's huge areas where that could use just knowledge and development, engineering, all these things. And so I hope that you'll all have the opportunity to participate in some of these areas, because I think it will be, moving forward, it's a necessary thing that we'll have to fight infectious disease going forward. It's also going to be an important part of, if we're going to move forward and be able to deal with cancer, I think a lot of it is understanding the immune system. But I'm not an immunologist. And so what I ended up studying was something that was going on inside of cells more than outside of cells. And the reason for that was only partially because it was too complicated for me, and partially because when we were doing those early experiments, we were doing them in what we call a model system, model organism. They're the little nematodes, they're earthworms, essentially, that are about 1 millimeter long. You could see them as a fleck of dust. And we worked on them because they were very simple to grow, and they were cheap, and they didn't really, nobody complained if we did experiments on them. They don't have major psychological things that we worried about. That may be a some people may have different views of that, but we didn't really worry that much about them. And so they were great organisms. They don't have T cells and B cells and antibodies in the same way we fight viruses. And so it turned out they have other systems which are much simpler but still effective. And so we ended up studying those for many years in that simple organism. And so the question is, if you don't have a flexible way to recognize the surface proteins of the viruses, is there any way you can still fight them? And to put a more specific question there, is there any way that you can recognize what a virus infection looks like as different from the normal things that a cell does? So think about, again, coronavirus. I hate, when I teach here, I now don't mention coronavirus at all, but so I'll apologize because it's too close to home, so to speak. But think about a coronavirus infecting cells. That's actually an RNA virus. And so it's a single strand of RNA, and it comes into cells, and then it makes a bunch more RNA, and that RNA gets translated through the normal cellular machinery to make proteins that do things. So in that whole process, most of it is just using natural mechanisms that the cell uses to express its own gene. There's RNA made, the RNA is used to make protein, the proteins then do things. And so it's a little bit difficult to have an underlying definitive mechanism which recognizes that that activity is unwanted. But there's one part of the viral life cycle that does let you get into that, and that's the replication of the RNA. So we don't replicate RNA, we replicate DNA. And then once the DNA is replicated, we make RNA from it. That's the transcription process. But the coronavirus or any kind of RNA virus needs to replicate its RNA. And so you need to have a copying of the RNA. And you might think that that was something that would be hard to catch for the cell. It would be a challenge for the cell to know, is an RNA replicating here? But there's a really great check on that. If you need to replicate RNA, then at least at some point, you need to have both strands of the RNA present, both the plus strand that makes protein normally and the minus strand where you created the replication. Because when you do replication of DNA or RNA, you start with one strand, you make a copy on the other strand. Those things are both just RNA molecules. So they don't have any sort of special properties, but together what they do during the replication process, you have a molecule that's got both strands, and we call that double-stranded RNA. And that's not part of normal gene expression. And so it turns out that a large part of our resistance to viruses that work through RNA is through the recognition of that double-stranded RNA molecule. Because it's not part of the natural gene expression, it becomes a signature, it becomes an indicator or signal that something unwanted is happening. And so as soon as the cell recognizes that double-stranded RNA, it's going to know, I should really deal with this. And so what's the best thing a cell can do under those circumstances? So if you were a cell and you detected that there was double-stranded RNA in your cell, and you wouldn't normally have that, how would you respond?
Well, probably dispatch the specialized cells that would annihilate this object.
Right. Yeah. So if you had an enzyme that could specifically cut up double-stranded RNA and not single-stranded RNA, because you don't want to destroy the cell's RNA, then you would be in good shape. So you need an enzyme that has specificity to the double-stranded RNA. And so there are those enzymes. My favorite one is called dicer. No negative things intended to the people who study a couple of other enzymes called RNase 3 and DROSHA. Those are also nice enzymes, but dicer is there. And one of the activities of the enzyme is it can chop up double-stranded RNA. So that all sounds good to me. So we have a cell that can get rid of double-stranded RNA by chopping it up. What's the virus going to do then? If we had that enzyme that was present in all our cells, wouldn't that protect us? Wouldn't we be free of viral infection at that point?
Well, I think the mutation will occur.
Right. But the good thing about the defense mechanism is that the virus, because it's a structure, a type of structure that we're going against, then a single change in the virus isn't going to get it out of that mechanism. So it's a lot more flexible than a mechanism that simply recognizes a specific sequence or a specific structure. It recognizes a whole class of structures that are intrinsically part of replicating. Well, that does do well for certain things, but of course, what viruses do, because they evolve quickly to defeat to try and defeat any mechanism, is a couple of things. One is that they will try and defeat the cellular mechanism, try and inhibit it as they infect. And that battle does go on back and forth, where there are viruses that encode inhibitors of the double-stranded RNA recognition and consequence machinery. So that's a part of it. But the other thing is that the virus can try and hide when it's replicating from the cell. It can try and do the replication in a little area where it prevents the cell from getting at that region. And so, so this was all known, that there were these double-stranded RNA nucleases, and that they probably could in part protect cells from replicating virus. What wasn't realized is the next stage in that, which is what happens in the real world if a cell detects a little bit of double-stranded RNA. Well, it chops it up into little segments, and those segments are then not going to be infectious because you've broken the information. If you broke all the information apart on your hard drive, you wouldn't really get much out of that, even though the information is all there in little bits. And so breaking up the information is good, but if you could only break up a fraction of it, does it help you? And so it turns out that one of the things that a cell can do with the little pieces of information is not throw them away once they're broken up, but use them beyond that. It saw a double-stranded RNA. It said, this is evidence of replication, although I might not be able to get my hands on all the replicating material. Let's take the little bits and use them as so-called guides. So we're going to take that information that you see, and now you know what to go after because you've seen it in double-stranded RNA. So if you can program an enzyme with those little sequences and use that to go around and get anything that looks like it, you'll be able to attack not only the replicating form of virus, but the form that's not replicating and trying to make more protein. And so that's this process called RNAi that, as I said, we kind of stumbled on working on gene expression in worms, but is a relatively general process. I should say it's not known at this point exactly what role it plays in the immunity that we have, because we do a lot of things with double-stranded RNA. It turns out that the first thing that happens if a cell sees a lot of double-stranded RNA is not that it triggers this very specific response. The first thing that happens is the cell just shuts down, because it says, if there's a lot of this, then we need to just take a break here, shut this particular cell down, make sure that there's not a problem. And if there is, we'll get rid of the cell rather than trying to have a virus replicate. And so that's one of the things that we do. But in the simple organisms, they use this general mechanism. And it turns out we have that mechanism also that's used somehow. And that's called RNA interference. So we have an innate non-specific response, and we have RNA interference, which is a specific response. And so all of these, in very similar to the extracellular response, the T cell receptor and the antibodies, all this is going on in cells all the time, trying to detect, trying to distinguish appropriate and wanted activity, which we want to let keep going, from things that are unwanted. And outside of cells, the kinds of cues that the cell has, the clues that it has are cells dying? Are they sick? And if they are, then the system, the immune system, knows to look for the components that are around and generate an immune response to them. Inside the cell, what it's looking for is double-stranded RNA and a few other things, single-stranded DNA in the cytoplasm, anything that sort of doesn't look right in terms of molecular biology, the cell will detect that and then try, first of all, to shut off what's there, but also to learn from what is present, to learn from the molecules that are there, and use that information to shut down anything that looks like it, to build its own sort of ongoing response to be able to turn things down. And so that is that I think, in my mind, that's sort of how the extracellular immune response, classic immune response, and the intracellular innate immune responses work. And it's been fascinating, and there's a lot to learn in this area. I mean, if it seems that I am a little confused in exactly how you'd recognize unwanted things, it's because we don't know all this, and there are many things left to learn and discover. So that's my 50-minute discussion of immunity and immunology, which I hope is helpful. But why don't I stop and take questions or anything on anything? Yeah, question.
Good afternoon. I have a question about the Spanish flu. What is your theory about H1N1 mutation into a safe form?
I mean, the flu is really scary, first of all. And, the degree to which the Spanish flu virus replicated, the speed to which it replicated, is really quite scary. So, I mean, I don't know how any of it happened, but I think we need to be ready for it to happen again, because in a way, I think that we thought we were in a very good position to deal with infectious disease. Maybe I learned in school that we were really done with infectious disease. We had antibiotics, we had vaccines against the most common viruses, and we were going to go forward and not have to deal with infectious disease. And that was wrong. That was a wrong thing that we were taught essentially in school. And it was wrong because human activity has changed, climate change, greater population, differences in behavior, all sorts of things lead to that. There's a much larger cauldron, there's a much larger ecosystem right now. And so if anything gets started, it really has a chance to expand much more quickly with us. And then other things are changing and viruses are evolving, and they have evolved over time. And so we're going to need to deal not only with COVID and with some minor things. AIDS was in between there. AIDS had characteristics which HIV had characteristics which allowed it to spread without being noticed in a population, and that was devastating. We will deal with other pandemics over time, and we really need ways to deal with them. And so, part of it is trying to make RNA vaccines are wonderful. The people that develop them should be greatly rewarded. I think that's tremendous. We're going to need to deploy that. We're not going to need to build those tools around a lot of different pathogens and build them quickly and thoughtfully. And we don't know how to do that exactly yet, but we're going to have to figure it out. Sorry, I'm slightly off topic. I don't know that much about the Spanish flu, except how darn scary it was. Yeah.
What's your opinion? So recently there were confirmed cases of the monkeypox. And how do you think, can it grow into the epidemic like with coronavirus?
Right. The answer, of course, is I don't know. Nobody really does. At its current level of transmission, I would still be much more concerned about tuberculosis, about COVID, about flu, other things too. But we haven't seen how the virus will evolve over time. And so I think that we should be building up our ability to respond to it, because the investment in a defense, which would be at this point making a lot more of the conventional vaccine against smallpox, which would work against monkeypox, that would be a valuable thing to have a lot more of at this point. And maybe we don't use it. There's also an economic issue with the whole thing, which is that we tend, our society, and I think maybe yours also, tends to work through the market. And the market says, if something is going to be useful, then definitely pay for it. If something might be useful, it's a little bit complicated to pay for it, because you have to get someone who's willing to take a risk to make a whole bunch of doses of some vaccine that we may never use, and also to be happy if we don't use it. And so we actually will end up paying for a lot of components that we don't use, just in the way that if you're trying to defend anything, you may have some resources that are spent in defense that hopefully you don't use. And that's counterintuitive until you need it, when you really want it.
Okay, thank you. Can you please then answer? You were telling us that when you learned about what will happen if, for example, you delete or copy some gene. So how you, I mean, practically perform this insertion and deleting?
Right, right. So there's a couple of ways to do it. This is the craft of genetics, and the really good way to do those experiments is actually doesn't involve any very fancy genetic engineering. If you take a species that you can grow in a laboratory and you simply select for the small number of organisms that say don't have fingers or don't have toes or have a better immune system or have more gut cells, and then you go and look at the genome and see what genes have changed, that's a really powerful method because you can get so many different kinds of changes. And so that's called a forward genetic approach, because you don't deliberately make changes in the genome. You simply assume lots of different changes are being made. And we use mutagens to increase that frequency of mutation. And then once you get those mutants, you pick out the ones that have interesting phenotypes, interesting properties, and you find what sequence has changed to cause that. And that's sort of a reverse way of doing the experiment. Other ways that people do it, at this point, people can put in a lot of double-stranded RNA to knock a gene down, or you can use recombination. The CRISPR system, as it's called, is one of the very nice ones to do that, where you break the genome in a specific place and then repairing it, you can put in extra sequences which can knock out a gene. And so there are tricks where using enzymology, using very specific protein RNA tools that can cut DNA, you again trigger a cell response that repairs itself. The repair can change a gene. And if you do that, if you program that properly, you can get all sorts of changes that you can then analyze the properties of. So if I knew the gene that I thought would be critical in a process, I can now manipulate it using something like CRISPR methodology. If I don't know what genes are involved in it, the very old method of classical genetics, where you just look for something that has the properties that you want and figure out what's changed in it, that's still the most powerful in forward genetics. It's been augmented greatly in recent years by a process called high throughput sequencing, where it used to take us years to find out if there was a mutation that had a given property that was interesting. It used to take years to find out what gene was affected, and now it takes a few weeks.
Thank you.
We had one question from the participant who is not here, left it at the Gmail newsletter. I'll try to wait a second. How could we use gene editing to first become carbon negative? For example, through making algae photosynthesis more efficient and then terraform Mars.
So the question is, can we use any of the methodologies that we've developed to make the world a better place beyond human medicine? I think that's probably the bigger question. And it's turned out with the one technology that I worked on a number of years ago, RNA interference, it has some applications in medicine, but they're a little bit limited because of the questions of delivery and efficacy. Those delivery and efficacy challenges mean that particular technology, if you apply them in a plant where they're very effective, you're not going to have a big problem. It's not going to make a big problem for people because the delivery and efficacy aren't very efficient. And so we don't care if we have a bunch of double-stranded RNA in our food. We basically have it all the time. And so if you know how to modify a plant, you can do it at this point. And I think one of the issues that's come up now repeatedly is this question of, should we be modifying plants? There's another question, should we be modifying the human genome? And if it's a sick person and their blood cells, I think everybody almost agrees that, yes, that's a good thing to do. But if you're modifying the germline, the next generation, that's a big question. But a plant, I don't have any ethical problems with, but you don't want to make a plant that's going to cause other environmental problems or going to cause other culinary or digestive problems. And so once we understand that, and we understand some of that because we've been modifying plants for years, we can try and make plants that are drought tolerant, try and make plants that are effective in doing their job in converting CO2 to water and plant material. We can do that more effectively if we understand how plants work and we can manipulate them. That means genetically modified plants are going to be part of our life. And I think that will happen over time. People will understand that in order to move forward as the human race, we can have concerns about any given genetic manipulation of our food, but we have to be aware that we're going to need it. Just the same way we're going to need to, if we want to have 6 billion people on the planet, we're going to need to be able to use modern medicine to make vaccines and things like that to protect us from things like COVID and pox viruses and other bacteria, other things. And so we will need genetically modified food eventually. I hope that the people embrace it sooner rather than later, because I think it is something that will benefit humankind. Another question, hand raised.
Okay, so is it true that the evolutionary improvement of like viruses, bacteria, is primarily because of humanity? I mean, we consume antibiotics and other pharmacological medicines, and so anyway, viruses find the way to get away from this, defense.
Yeah, I mean, it's a good question. Is it all about people? And I think people are one of the drivers of changes in the environment currently. And so I think probably we're a little bit of it. But there would be viruses evolving even if there weren't people doing dumb things at this point. I think that's true. And if you look at we sort of see a fossil record of the viruses that have been around through genomes, because the DNAs occasionally DNA viruses particularly, and sometimes RNA viruses get into genomes and get carried along for millions of years that way. And so we can see that there's been evolution over time and well before humans had quite this control of the environment. So I actually think we're not the only source of problems. And yet as we accelerate climate change, as we accelerate environmental change on the Earth, that will certainly become more prevalent. There's going to be more opportunities for viruses to build a larger area. And the first part of it is our climate change, and part of it is just that we have made travel and exchange between geographic areas so much more efficient in the last 100 years or so. That has also increased the transmission of disease around the world and increased the risk. So it is accelerated to some extent by human activity, but I don't think that it is only because of human activity. If the world were full of squirrels, we'd still have a lot of viruses coming up and causing trouble.
Good evening. We talked a lot today about viruses like flu, coronavirus. So can you give us, like in general, your prediction? Does there any good chances in the nearest future to eliminate flu or coronavirus, like we did with tetanus or polio, for example? Thank you.
I mean, it's a really good question. I'm not a card-carrying viral, epidemiologist. I think it may be hard to eliminate coronavirus, and I say that we've had some pretty good tools that have come up over the last 2 years. It's really difficult to deploy the tools effectively. Part of what happened, so a mask is a really good tool, and it's not that it would have totally stopped the pandemic, but they would have saved millions of lives probably. But people don't want to wear them, which is fine. Some people, though, will begin to spin stories that the masks are harmful. Vaccines are not perfect against any virus, but they can really help. And they would have helped in the pandemic. We could have been a little bit better in distributing it, and I hope we will be as we go forward, and resources for that will become available. But the other problem is that there's a certain component of people who, not with any great, because there's beneficial debate about whether any vaccine is safe, but there are people who have tried to build their own professional branding or personal branding on essentially spreading misinformation about vaccines. And that doesn't help. And that's made that tool less effective in places where there could have been 80, 90, 95% vaccination. It's way down because some people believe the stories that there's a microchip in there. If you think that the vaccine might not quite be effective and you're deciding whether to use it, that's fine. But if you think there's a microchip in there, or that it's going to turn you into something else, then I think that's a problem. Or if you think that many, many people have suddenly died because they took the Moderna vaccine, that's not true. And people are dying because they have those misimpressions. And so I think that we do have that problem. Now, that being said, there have been amazing successes in infectious disease. Polio is not completely wiped out, but it has been greatly controlled through good work. HIV is in no way wiped out, but there have been some amazing advances in 40 years in HIV research, and then bringing that into the clinic and making the treatments, frankly, accessible to a lot of different people. And that's been a huge example of how to make it advance. And the people at all stages who made that work have been tremendous. And again, at the same stage, there have been people who have injected misinformation at the cost of some control of the pandemic, and certainly at the cost of lots of lives. So I think there is the potential to control COVID, but I suspect that given its ability to mutate and given some inability, we can't force people to do things, and that's probably fine. But I think that the combination of the viral mutation and human misinformation means that probably the best we could do is that we'll have much better tools to deal with it. We will be able to turn it into what's called an endemic virus, where it's here but problematic. But it's going to be my sense is it will not be something that will be just a few years. It'll be in previous years, you had to worry about the flu, you had to worry about bacterial infections, all sorts of things. This will be one more thing to worry about, not of the same scale as HIV or as COVID was in 2020, but at the level to which it's one more thing you worry about if you're traveling in the winter.
All right, thank you very much. I guess probably that's, we don't have enough questions, I guess. Thank you for coming.
Thank you very much. And as I said, all do well, go forward, make changes, learn science, make a difference in the world.
Thank you very much. Have a good day.
Right, take care.